Namaskar. This talk is about multifactorial inheritance. This mode of inheritance is really a concept, and lot of things are not clear or as exact as they are for monogenic disorders or chromosomal disorders. When we think of any disease, probably except trauma, almost every disease has some component of genetic etiology, uh, and we can say that probably susceptibility to that disorder is always inherited in some genetic fashion. When we think of many disorders, and we can categorize them differently. So at one end, there are diseases like Duchenne muscular dystrophy and spinal muscular dystrophy, where there is a disease causing mutation in the patient, then the patient will always have that particular disease. While same thing we talk about and say that osteogenesis imperfecta and hemophilia and PKU are monogenic disorders, but we know that for hemophilia to bleed or for osteogenesis imperfecta patient to have fractures, there may be minimal trauma required as an environmental component. Same is true for phenylketonuria and galactosemia, unless the patient with these disorders gets some milk products which contains amino acids, proteins, or in case of galactosemia, galactose, till then the individual will not get a particular manifestations. So these are mostly on one spectrum called as monogenic disorders with variable amount of environmental component, which is usually small. And on the other hand, there are other disorders like infectious diseases, which we call tuberculosis or HIV. There also we know now that there is susceptibility to these infections is well documented. In between are the many common disorders like diabetes, hypertension, etc., psychiatric illnesses, and many congenital malformations where a multifactorial etiology is thought of, and there is research going on to identify genetics etiologies. The difference between these monogenic disorders and multifactorial disorders is that multifactorial disorders are quite common in general population, and usually they have low risk of recurrence in the family members. When I told that HIV is also a genetic disease, you might be surprised. But you can see these particular screenshot of in Mendelian inheritance in man, which shows that various genes which are known to be associated with resistance to immunodeficiency HIV virus. This was identified that a group in African countries, they have resistance to AIDS, and the genes were identified because of that. So HIV AIDS as a prototype of multifactorial diseases, you can see that there are many viral factors like viral load, virulence, as well as many factors for the patient like age, sex, other disorders, etc., nutrition. And on the other hand, genetic factors in the form of mutations in genes, mainly called CCR5, are very important for development of AIDS. Because this particular gene, a protein, is required for the virus to get entry inside the cell. The information about this particular genetic variant, which gives rise to susceptibility to AIDS, is used for the treatment in the form of developing using inhibitors for CCR5 or editing this particular gene by genetic techniques so that the HIV can be treated by gene therapy. You might have heard about of genome editing of a fetus for development for so that they are resistant to HIV in a specific country, and this has been reported. So, similarly for tuberculosis, if you go to online Mendelian inheritance in man, there are a list of genes which cause susceptibility to tuberculosis. And here on the other side, I'm showing the picture of Victor McCusick, who is the father of this particular catalog of online Mendelian inheritance in man. Initially, he used to he made a book for this catalog in the book form, but gradually the numbers entries increase so much that now it is only online database freely available for everybody. Please remember that photograph of Victor McCusick, because I'm going to tell something interesting about him in further. Multifactorial disorders, as I said, they are very common when you see as compared to the general prevalence of single gene disorders or chromosomal disorders. And that is why these are very important areas of research, and as they affect large number of population. Examples I have already told about multifactorial disorders, some congenital malformations, especially neural tube defects, cleft lip, and pyloric stenosis, and mainly almost all adult onset diseases like diabetes, epilepsy, glaucoma, psychiatric illnesses, age-related macular degeneration, ischemic heart disease, etc. How we decided that these disorders have genetic etiology? Because for many of the disorders, genes were not causative genes or susceptibility genes were not identified previously, and still they are in the preliminary stages. But what was observed, that the risk of these disorder amongst the siblings or family members of an individual with this disorder was increased as compared to the prevalence of that particular disease in the general population. So you can see that the risk of psychiatric illnesses is increased 7 to 10 times in the siblings of an affected individuals versus just general population. So Crohn's disease and psychiatric illnesses, they have a significant genetic component in the etiology as compared to the other disorders uh, seen in the table. The heritability of each disease is also known for many disorders. And as I said, for schizophrenia and asthma, it appears to be higher as compared to the other disorders like hypertension and malformations. So how does one calculate heritability of a particular disorder? This is mainly by twin studies. So if you have data about concordance of a disease in monozygotic twins versus trizygotic twins, you can make some inferences, because monozygotic twins have all their genomes exactly similar, and naturally if a disorder has more genetic component, the possibility that the both of the twins will get the same disease is higher as compared to both of the dizygotic twins getting the disease. uh, this is data about type 1 and type 2 diabetes in concordance rate in twins. And you can see that type 1 diabetes has more genetic component, because the concordance rate is more as compared in monozygotic twins as compared to the dizygotic twins. The heritability, hence, for type 1 diabetes is more than type 2 diabetes. The other important way to study is to look at twins which are reared apart. And you can see there are very interesting work being done for that. And their research showed that the twins were similar in their abilities and personalities despite the extreme differences in their rearing circumstances. So the twins which were separated during early childhood, and if they were reared apart, then still their behavior continues to remain the same. And here you can see I had shown Victor McCusick. So he had a twin, and these are their childhood photographs. And then at a later age, during old age, so the authors say that look at their hand positions and look at their legs, the way they are sitting and their hands are kept. So these still continue to, they have the same similar um, posture is there. And this suggests that probably behavior and lot many things have genetic component in the etiology. The concept, what we have observed, that many the risk of the disorder in this family members of an individual with multifactorial disorder is more. And the hypothesis for that is that probably those individuals who have a multifactorial disease, then the genetic susceptibility genes and there are mutations in them, the load of mutations in their genetic susceptibility genes is higher, and hence probably those genes mutations will be shared in the family members as well, and hence probably the risk of disorder of these multifactorial disorders in the family members is higher in the relatives. This has been identified by various studies. So the simplest form of what I can give an example is that if you consider a disorder like maybe diabetes mellitus type 1, so the individuals with a normal or wild copy of a gene are represented by yellow color, while those with mutated copy or the different variant are depicted by blue color. So you can see that those with a variant in that particular gene, maybe gene A, are more in patients with diseases as compared to the control normal population. So this indicates that the possibility of getting a disease is more if an individual has a variant in gene A. Of course, it doesn't mean, you can see that there are many individuals without that variant and still they have got a disease, and also there are many individuals with the same variant in the normal population and not getting the disease. So the message is that these association means that the possibility or risk of getting the disease is increased as compared to the risk in those individuals who do not have that genetic variant. Look at this another data. Here you can see the same variant. It's present equally in the normal population as well as patients with the disorder. And this means that this variant is not associated with that particular disease in concern. So such type of association studies for one gene by one gene were being done depending on the possibility or some hypothesis about candidate gene, like for MTHFR, genetic variants and neural tube defects and so on. But with availability of genome wide testing, there are many studies where thousands and thousands of patients and controls were studied for thousands of genetic variants all over genome. This can be done by SNP microarray, which has all variants on its chip, or it can be also done now by next generation sequencing based methods like whole genome sequencing or exome sequencing. So the advantage of this is that there is no candidate gene hypothesis. So we do not know what is the cause, what is that particular reason that particular gene may be associated with the disease. We do not know the pathogenesis, anything. But just strong association can be looked for in this huge data of patients and variations by computerized work and applying lot of statistics. And one can look at those various genetic variations which are there, almost 1 per 500 base pair there is a genetic variant. So one can look at and find out whether such type of variants are associated with the disease. This has been very successful in a disorder called age-related macular degeneration, which is a cause of old age blindness, a very significant cause. And you can see that the prevalence increases with age, but prevalence varies in different populations, and that must be based on their ethnic variations and genetic variations. So what these genome-wide studies studied for age-related macular degeneration identified sequence variation in a gene called complement factor H polymorphism. So you can see this line shows cutoff, and you can see these the variations for age-related macular degeneration, which were important, identified, and their p-value was very significant. So those individuals who have homozygous variant in this particular CFH gene, the risk allele, the likelihood of developing age-related macular degeneration is increased by almost 7 times. So this means this particular variant is an important etiology or for the susceptibility of developing this disorder. Of course, simultaneously, there is a very well accepted and very well known environmental factor for this disorder, and that is smoking. Such type of studies are done in thousands of individuals. Like this is a data of 30,000 individuals, and you can see that in addition to this CFH gene, some other genes like ARMS2 and etc., they are also identified as causative variants and contributing to the genetic etiology of age-related macular degeneration. In addition to these two, the many genetic variants were identified, and you can see they are identified in all over genome because the technique used was genome-wide association study. Uh, once these disorders, once these genetic variants are identified, one can look at them in different population. As we saw that many, the susceptibility varies from population to population. And in our Indian data, we also studied variations for in CFH gene, ARMS2 gene, in age-related macular degeneration. And you can see the odds ratio of with the CFH association was almost 3.36 and reasonably significant. But when you see these very many variants, other than really ARMS2 and CFH, which are here the topmost, the contribution of other variants is usually much less as compared to these two particular variants. And naturally, as I told you again, such type of genetic susceptibility genetic variants cannot be used for diagnosis because they will be present in normal individuals as well as they will also be present in patients, and many individuals without the variants also will get the disease. So what is the use if we cannot use them in clinical practice? The use is understanding, trying to understand the pathogenesis of the disease. So many of these genes which have been identified are into the pathway of cell migration, cell proliferation, apoptosis, immune function, etc., inflammatory markers. And naturally, based on this understanding of pathogenesis, the various treatment strategies can be developed for such type of complex multifactorial disorders. So that is the objective of identifying genetic susceptibility for these multifactorial disorders. The development of a particular disease in an individual ultimately will depend on the balance between the number and function of susceptibility genes versus genes which provide resistance or help in not developing the disease. So good and bad genes plus environmental factors. So such a combination is too complex to be really used for clinical purposes. The other issues in multifactorial diseases is that the phenotypes are all extremely heterogeneous. Even so-called type 2 diabetes, you take, there are many different types of diabetes, and there may be different types of diabetes and susceptibility genes in different families. There are many monogenic diabetes also, maturity onset diabetes of young. Same way, some monogenic forms of hypertension are also there. Monogenic hyperlipidemia genes are there, which contribute to ischemic heart disease. And there is a significant component of environmental factors. The multiple genetic variations, so it's not very easy to dissect out which variant will give rise to the disease and how, what is the possibility. That is a really challenging thing. Also, the various calculations using this information about variations in the families uh, will change depending on ethnicity and different populations. For research, the phenotype is are also very difficult, because for psychiatric illness, the labeling the individual with a definitely having the disease versus normal variations, so many times the boundaries are little blurred. The number of patients and controls needed are very, very high. Um, Basically, when we do such whole genome-wide studies, you get associations, and these associations later one needs to work on them to understand if there is the biology, what is the biology behind it. But just association means nothing, as it does not provide any information about the causal nature of those variants. The type 2 diabetes mellitus is very worked up for the genetic etiologies. More than 150 loci have been identified. Um, then collectively, whatever the information about genetic susceptibility loci is identified, it explains only 10% of overall disease risk. So naturally, then there must be many more genetic susceptibility as well as environmental factors which we still don't know, and really we cannot use this type of genetic information for patient care. For patient care, if many family members, if they want, we can have information which is observed in the population, and these are called empiric risk data are there, that if there is one individual with diabetes, then the risk to his siblings is 10%, and so on. So such type of information, if both the parents have diabetes mellitus, then there is a possibility 70% for developing diabetes in the offspring. So this type of information is not very important for diabetes and complex disorders late onset, but this is very important for congenital malformations. Neural tube defects is the commonest congenital malformation and is seen in 5 per thousand births. So for a family, if there is one child with neural tube defect, then the risk of recurrence in the siblings is 5%. If there are two affected children with neural tube defects, then the risk of recurrence is 10%. And though the genetics is not yet well understood, it has been seen that periconceptional folic acid really helps and decreases this risk of recurrence significantly. So it means, though we are not definite about the etiology, the therapy of periconceptional folic acid some way helps and is taking care of either genetic susceptibility or some environmental factors. The lastly, I would show this picture from the screenshot from the website of National Human Genome Research Institute, and here they are showing that these are the individuals at one end who have some genetic variations, and they are at high risk of complex disorders like ischemic heart disease, etc. And one can try to identify and look at their genetic data to identify what is the possibility of getting that particular individual the disorder. And this type of calculations are being done and called as polygenic risk scores. But again, to remind that as compared to the monogenic disorders, if the individual has one sequence variation in a gene, the possibility of developing disease is 90 to 100%. While in case of multifactorial disorders, you are going to look at many variations in many genes, and it will give some possibility, and whatever we get is just possibility of increased risk over and above general population, and not, it's not definite. So naturally, such type of polygenic risk scores, they have limited utility in predicting whether the individual will develop disease or not. And because they are not very certain, they cannot provide any certain information, certainties are not there. So polygenic risk scores, hence, though are in research mode, people are trying to find out if they can be clinically useful, but they are not used for by health professional at present, and there are no guidelines for practice. And researchers are still trying to improve how these scores can be generated, whether they will be applicable from one population to other population, and so on. And they are only associations and no causal relationship is there. The intervention suggested after, if this polygenic risk score shows that the risk for ischemic heart disease is more, the interventions are lifestyle modifications, and that type of advice is also possible just by taking family history, BMI, and lipid profile. Hence, American Society of College of Medical Genetics and Genomics have taken out their statement, and they say that even with the most sophisticated technologies available, genetic studies on complex traits and diseases constitute an inexact science and do not identify affected individuals as it does for monogenic disorders. So, in most instances, this is not a part of general clinical genetics practice, and in this context, the emergence of direct to consumer testing for these type of complex multifactorial disorders is particularly troubling. So I would end by telling that we need to know about keeping ourselves updated about research in these multifactorial disorders, but at clinical practice and in clinical situation, their utility is very limited.